The human body is a marvel of biology, constantly adapting to the passage of time. But as the years go by, changes in metabolism, hormone levels, and muscle mass can lead to frustrating shifts in body composition, most notably, the stubborn accumulation of fat around the midsection. Now, a groundbreaking discovery from researchers at City of Hope offers new hope for understanding and potentially reversing this age-related transformation. Rather than being a fixed outcome of aging, the rise in belly fat appears to stem from a newly identified biological mechanism that could be modified or even prevented with targeted therapies. The scientists at City of Hope, working in collaboration with UCLA, have identified a previously unknown population of stem cells that emerge in middle age and drive fat accumulation in the abdominal region. These cells, called CPS, short for committed preadipocytes, age-specific, appear to be activated by aging itself. Unlike most adult stem cells that slow down with age, CPS ramp up in activity, producing new fat cells at an accelerated pace. This process helps explain why many people experience a noticeable increase in belly fat during their 40s, 50s, and beyond, even without significant changes in diet or exercise. Crucially, this discovery does more than just identify a problem. It opens a new door to possible solutions. At the heart of this process is a signaling pathway known as LIFR, leukemia inhibitory factor receptor, which acts as a kind of switch that turns on CPA activity. In young individuals, this pathway seems dormant or unnecessary for fat production. But in older bodies, it becomes a central driver of new fat cell creation. By targeting this pathway, scientists may be able to selectively dial down belly fat production without compromising essential fat stores elsewhere in the body. The significance of this research extends far beyond aesthetics. Visceral fat, the type that accumulates around abdominal organs, has long been associated with increased risk for serious health issues like type 2 diabetes, heart disease, and metabolic syndrome. Reducing this fat, particularly in older adults, could translate into longer, healthier lives. This isn't just about looking slimmer in the mirror, it's about transforming the trajectory of aging itself. Encouragingly, the findings have already been validated in human tissue. Using advanced techniques like single-cell RNA sequencing, researchers identified similar CPA cells in human fat tissue and observed a clear increase in their presence as people aged. These human CPS, just like those found in mice, were highly active in generating new fat cells, reinforcing the biological connection between aging and abdominal fat gain. This line of research also offers a more optimistic perspective on the aging process. Rather than viewing midlife weight gain as an irreversible fact of life, it reframes it as a solvable biological challenge. With further investigation, it may become possible to prevent or even reverse these changes through precise medical interventions, whether through drugs that block the LIFR pathway, lifestyle strategies that reduce CPA activation, or other approaches that haven't yet been imagined. The road ahead is promising. The next phases of research will delve into how CPS operate in the human body over time, how they can be tracked or modulated, and how new treatments might target them without unwanted side effects. These efforts could redefine how we approach metabolic health in middle and older age, shifting the focus from reactive treatment to proactive prevention. This discovery also underscores the importance of advanced, age-specific research into fat biology. For too long, obesity has been treated as a monolithic issue with one-size-fits-all solutions. But aging bodies operate differently. As this study shows, fat accumulation in older adults isn't just a matter of eating too much or exercising too little. It's rooted in cellular changes that can now be studied, understood, and potentially controlled. In the grander scheme, this marks a profound step forward in our understanding of how aging affects our bodies and opens new avenues for helping people maintain vitality, energy, and health as they grow older. The identification of CPS and their connection to the LIFR pathway gives science a powerful new lens for looking at obesity, not as a permanent sentence, but as a dynamic process that can be shaped by knowledge and innovation. And in that, there is real reason for hope.